Hello, everyone. My name is Advani Garrido, and today I'm going to be presenting a research study that I looked at repurposing neuroactive drugs for the treatment of glioblastoma. The paper I chose is titled High Through Throughput Identification of Repurposable Neuroactive Drugs with Potent Anti-glioblastoma Activity. And it was published by Lee and colleagues in Nature Medicine in 2024. So, I chose this study because glioblastoma is a very aggressive brain cancer and treatment options are still pretty limited. What I found especially interesting in this paper and about this research is that instead of developing a completely new drug, the researchers looked at medications that already exist and asked whether some of them could potentially be used against a glioblastoma. One drug that really stood out was virotoxetine, which is actually an antidepressant. Also, um, glioblastoma is an aggressive form of brain cancer. So if we move on to slide number 2. So, glioblastoma and the clinical challenge. So, before getting into the actual study, I want to give a little background on why glioblastoma is so difficult to treat. Glioblastoma is a highly aggressive primary brain tumor. One of the major challenges is that it can be very difficult to completely remove through surgery. So, even after surgery, patients usually still need additional treatment. The standard approach typically involves surgery followed by radiation and chemotherapy. But even with these treatments, reoccurrence and treatment resistance are actually still very common. So this is really where the importance of the study comes in. We have already have treatment available, but they don't always provide a long-term control of the disease. So because of that, researchers are looking for different strategies that could either work on their own or potentially make the treatments we already have more effective. So, what was this goal? So the main question that the researchers want to answer was, can existing neuroactive drugs be repurposed to effectively target glioblastoma? So instead of starting from scratch and trying to develop an entirely new medication, they screened drugs that already have known biological effects, and they wanted to identify which drugs showed the strongest anti-glioblastoma activity. And then they investigate what those drugs were actually doing to the tumor cells. So, another important part of the study um, was actually that. Wait, 1 second. Yeah, so sorry, I lost my train of thought. So, another important part of the study was looking at whether these drugs could potentially work alongside glioblastoma treatment. So, the goal wasn't just to find a drug that kills cells in a lab, they wanted to find something promising enough that could potentially be investigated further as part of a glioblastoma like treatment. So, the study approach. To approach this question, the researchers started with glioblastoma samples from 27 patients. So, they then performed a large drug screening. Overall, 132 neuroactive drugs were screened, and they analyzed more than 2,500 individual drug responses. This is important. Why? Because glioblastoma tumors aren't all exactly the same. A drug could work really well against cells from one patient, but have a different response in cells from another patient. So, using patients' derived samples gave the researchers a way to look at something that has variability. So, after screening the drugs, they ranked their effectiveness and identified the most promising candidates. So those candidates were then investigated further to then understand their effects on glioblastoma cells and were tested in additional environmental models. So basically, they started a very broadly with a large drug screen, and then gradually they came down and narrowed it down to the drugs that showed the most potential. So drugs, drug screening identified promising candidates. So this figure, the one right here, shows one of the major results from the screening. So the researchers screened 132 drugs total. This included 67 neuroactive drugs and 65 oncological drugs. They then ranked the drugs based on their activity against glioblastoma cells. So on this graph that you see right here, the drugs toward the left side had higher PCY scores, meaning that they showed a stronger activity in the screening. However, something interesting is that several of these neuroactive drugs actually performed very well, even though these medications weren't originally developed as cancer treatments. The responses also varied between patient-derived samples, which again shows how complex the glioblastoma can be. But one drug that emerged as a particularly promising candidate was the virotoxetine. You can actually see it highlighted towards the upper left portion of the right here in pink. You can actually see it highlighted towards the upper left portion of the graph. So because of the activity they observed during the screening, and this during the screening, and then the researchers selected virotoxetine for further investigation. So, why did the virotoxetine actually stand out? So now the question um, becomes, why is it so interesting? Why is this drug virotoxetine so interesting? So virotoxetine is already an FDA approved antidepressant. Obviously, an antidepressant and a cancer treatment sound like two completely different things, which is part of what makes this finding super interesting. The researchers found that veritoxetine showed potent activity against glioblastoma cells. They then looked more closely at what was happening inside the cells. So one important finding that was that veritoxetine triggered a rapid influx of calcium into the cells. This was associated with cellular stress responses and changes connected with reduced tumor growth and cell death. The figures here are showing some of these calcium-related responses after treatment. So the researchers weren't only seeing that the glioblastoma cells were responding to that drug, they were also beginning to identify biological mechanism that could actually explain why help explain why. And because veritoxetine is already an approved medication for another purpose, it became that much more interesting from a drug repurposing perspective. So veritoxetine plus standard chemotherapy. So after seeing these results, the researchers also want to know whether veritoxetine could work alongside treatments that are already used for these glioblastoma. They tested the veritoxetine with temozolomide or TMZ and lomustine, which is also called CCNU. So right here, these ones. So this survival curve right here shows one of the most important findings again from the study. So instead of only looking at what happened in cells in a dish, the researchers were looking at survival in a preclinical model. So each of the drugs alone significantly prolonged survival, but when veritoxetine was combined with standard chemotherapy treatments, they observed an even greater survival benefit, even greater survival benefits. So if you look at this curve right here, the combination groups extended farther to the right, showing that survival was prolonged compared to the control groups, as you can tell right here. So this is important. Why? Because veritoxetine wouldn't necessarily have to replace the treatment patients already receive. The possibility is that it actually could be eventually investigated as an add-on to treatment that we already have and that enhances the effects of the standard therapy. Of course, these are still preclinical findings, so this doesn't mean that veritoxetine is currently an established glioblastoma treatment, but it gives a strong reason to further investigate it. So, study outcomes and significance. So, overall, I think one of the biggest outcomes of this study is that it demonstrates the potential of drug repurposing through high throughput screening. The researchers were able to identify unexpected anti-cancer activity in drugs that weren't actually originally designed to treat any cancer, which is crazy. So, in this case, veritoxetine emerged as one of the most promising candidates. Drug repurposing can actually be very interesting approach because we're looking at the existing medications that we already have from a completely new and different perspective instead of always starting with a new compound and a new treatment. At the same time, it's important to not overstate what the study found. These results are promising, but additional clinical research is still needed before we can determine whether Vitoxetine would actually be safe and effective as a glioblastoma treatment in patients. So, I would say that the major outcome isn't that we've suddenly found a new treatment for glioblastoma or a new way to form glioblastoma. It's that researchers identified a very promising direction that deserves further investigation. So, further research and take home message. Going forward, there are several questions that still need to be answered. So, researchers need to continue evaluating the safety and effectiveness of this approach and determine what therapeutic dosage or dosing would actually be appropriate in this cancer setting. They would also need to investigate which drug combinations are the most effective, especially because this study showed promising results when Veritoxetine was combined with standard treatment. So, most importantly, clinical studies in patients would be needed to determine whether the benefits seen in this experimental model actually translates to humans. So, yeah, but take home message. So, the main take home message from this study is that medications may have useful effects beyond the purpose that were originally developed for. And antidepressants showing anti-glioblastoma activity is a really good example of that. Glioblastoma continues to be extremely difficult to treat, so looking at existing drugs in a new way could potentially give researchers like another direction for developing these treatments. Overall, this study shows that drug repurposing, especially like the neuroactive drugs like Veritoxetine, could be actually promising for future glioblastoma research. So this was, and then now this is the reference of the PDF and the paper that I researched on. I hope this gave you a little more information, and thank you so much.